Andrea, you and I work together in the European Institute of Oncology in Milan. Welcome to Birmingham, to the National Cancer Research Institute. You're obviously in the top 1% of cancer researchers, otherwise you wouldn't be asked here. So there you go. Tell me what you were talking about today. You're a mitotic man, you're a spindle man. I know that, but tell us about it.
I've been talking about how cells progress through mitosis. And sort of trying to convey the complexity of the process. Is a process that has to do with dividing chromosomes, that is the DNA material, into two equal parts that will be given to daughter cells. The process is clearly a very interesting process from the perspective of targeting cancer cells, because sort of the idea is developed over the years that you can sort of create or set up an ambush to cancer cells as they go through mitosis. They need to go through mitosis as much as normal cells, but they probably rely on mitotic sort of defense mechanisms, or mechanisms that allow cells to divide chromosomes properly more than normal cells. And so sort of historically, the taxanes and the zinc alkaloids have been sort of used as anti-mitotics to...
Historically being my vintage.
Right,
40
historically
means,
for many years now. And the pharmaceutical industry has become more and more sort of progressively interested in other targets besides the targets of the taxanes and zinc alkaloids, which are the microtubules, in other targets that are important for mitosis.
Such as?
Well, such as some so-called kinesins, which are molecular motors, motors that transport things over microtubules, or the spindle assembly checkpoint components, such as Aurora B kinase, or molecules like polokinase, which are important for forming the mitotic spindle, the structure that sort of is devoted to parting the chromosomes. Now, sort of apparently, these seem like sensible targets for killing cancer cells. The clinical studies so far have been quite disappointing, and it is clearly crucial to understand why. Yeah. What are the problems? Are these not the right targets? We're not targeting them properly with the right dosage. Is combination therapy with other chemotherapeutics like Taxol going to work better than sort of providing sort of these compounds in isolation? So these are open questions. In my perspective, what is particularly important is that we understand better the mechanism of action of these compounds. And therefore, sort of we've been trying to collect these compounds, those that are available for laboratory use, so whose usage is not restricted by companies, to do what is called chemical biology, essentially sort of try to understand exactly how these drugs work in the cell, what exactly are their targets, and the mechanism that drives cells out of business, essentially. And so this is where we've been investing the most.
And what's come out?
Well, sort of what's come out is quite interesting, and that is that we've been, in particular, sort of trying to clarify that certain kinases, so some targets for carrying drugs, do things that have not been recognized in the cell. And so, for instance, Aurora kinase has not been recognized as a component of the spindle assembly checkpoint, whereas our studies demonstrate that this is clearly the main engine of the mitotic of the spindle assembly checkpoint. And therefore, we think we need to understand the mechanism of action of these drugs to sort of predict how they will work. So this is a major outcome of our research.
Do you envisage these drugs eventually getting more refined or used in a different way, for instance, with taxanes?
Right, I think a combination therapy may be particularly sort of important. It's worth trying. And I think the other thing that needs to be done is to understand exactly what the targets are. What are the best targets for mitosis? Do we really know that the aurora kinases or polykinases are the best targets? They were identified initially because they're overexpressed in cancer, many tumors. This is not necessarily a good reason for choosing a target, and if we had a more rational approach to the problem, maybe we would identify new targets. Now, there's been clearly a lot of progress in that respect. A lot of this goes through running screens that are appropriate to cancer phenotypes, and in particular, for instance, the group of David Pellman at the Dana-Farber Cancer Institute ran a beautiful screen that led to the identification of certain targets, new targets, that may be particularly relevant for cancer cells. And so sort of I think more screens and the identification of new targets that are literally specific to cancer cells in mitosis might improve the situation.
And they have to be targets which are lethal, which cells cannot, develop resistance pathways to.
Right.
As a clinician, that's the preference.
Right. So the obviously, sort of what one would like ideally to do is to find to address a target that is good from the perspective of what is called synthetic lethality. What it means is having a target that is specific to cancer cells, not to normal cells, because cancer cells have lesions. They make them susceptible to the usage of these specific drugs.
Good luck, Andrea. Thank you very much indeed.
Thank you. Thanks.